give you guys an introduction to our first speakers. I'm sorry, here it's listing Dr. Thomas Green, but it's also Dr. Fernanda Laezza, I think it is. I forgot, don't know how to pronounce it. So let me start with Dr. Thomas Green. He received his PhD in experimental psychology from the University of Kentucky, and then completed his postdoctoral training for the University of Texas at Southwestern Medical Center with Dr. Eric Nestler. He is currently an associate professor at the Department of Pharmacology and Toxicology at the University of Texas branch, University of Texas at the medical branch, excuse me. And his co-presenter today and co-recipient of the R03 is Fernanda Laezza. She received her medical training at Torino, Italy, and then continued on to get a PhD in neuroscience at Emory, studying brain disorders with genetic and environmental aspects. And today we'll be hearing some of their work where they are working on preclinical animal models, focusing on the molecular mechanism underlying the protective addiction and depression phenotypes that can be used in order to treat addiction, depression, or anxiety. And here we will be hearing some of their work on the understudy target of GPR12, and with the title GPR12 in the nucleus, accumbens and addiction, and electrophysiology and behavior. I'll hand it over to you.
All right, let's share my screen. All right, you can see the PowerPoint.
Perfect. Yeah.
Okay, so first I want to say thank you for allowing us to present here. This is a project we're really excited about, and this is a program, the IDG program is something we're really excited about. I think we're a great demonstration of why it's important, because we've been excited about this target GPR12 for about 7 or 8 years. We've included it in some R21 applications. I think we submitted 4 applications, never got funded, and the reason they never got funded, even though they're an R21, they never got funded because these are new targets that people haven't studied before. The premise is not as strong as it would be if these were targets that people had already studied, and so we think that the R03 mechanism through the IDG is something that leveled the playing field so that we were able to get this funded, and so we're super excited about this and excited to participate. Myself and Dr. Laezza. Dr. Laezza is an electrophysiologist, so the electrophysiology data you're going to see, she's going to present that, and I'm going to present the behavior data. So this project is based on this idea of regionally enhanced gene expression. This is a real simple idea, is that genes, by and large, don't get expressed in cells where they're unnecessary. Part of that is through the last maybe billion years of evolution. Many, if not most, of our ancestors were on the verge of starvation, and making genes in cells, making proteins in cells where they're unnecessary, it wastes energy, and it takes up nutrients. And so there's over 1,000 proteins in the proteome that are involved in gene expression, and people always focus on the ones that increase gene expression, like transcription factors, but there's a lot of, I think, an understudied, more important aspect is limiting gene expression in cells that where it's not necessary. And so I noticed a long time ago, from looking at the Allen Brain Atlas, I've noticed that genes that I had been studying in relation to addiction, depression, and anxiety tended to have pretty selective expression in the nucleus accumbens shell, ATF3, serotonin 2C receptor, CART. And so the idea is that other genes that are expressed in the nucleus accumbens shell, the shell is involved in addiction, depression, and anxiety, other genes that are expressed in this area selectively. There are no genes that are perfectly expressed in the accumbens shell and nowhere else, but there are genes that show enhanced expression. And so we wanted to investigate that. The Allen Brain Atlas is one of the most incredible resources that's been developed for neuroscience. Basically, they did in situ hybridization, looking for RNA expression in brain slices of mice. And they did in situ hybridization for every gene that was known at the time. And so they developed this massive database of expression profiles, and you can see some of the expression graphs here for some of the genes that I've looked at in the past. And so it gives us the opportunity to do regionally enhanced gene expression using the Allen Brain Atlas. And so we discovered about 178 genes that showed enhanced expression in the nucleus accumbens shell. We wanted to do a convergent functional transcriptomics study where we take data from multiple data sources and intersect those data to come up with novel targets. And so we did traditional RNA sequencing for the nucleus accumbens, looking at environmentally enriched versus isolated animals. We know that environmental enrichment produces a protective phenotype for addiction-related behavior in rats. These enriched and isolated animals were self-administering either cocaine or saline as a control. And so we have genes that were regulated by cocaine, genes that are regulated by environmental enrichment, and genes that show enhancement in the nucleus accumbens shell, which is important for addiction-related behavior. Most people who do these convergent approaches do it at the gene level. However, these data sets have such massive type 2 error that you can lose so much by doing it at the gene level. So we did our analysis at the gene set level. So we did ingenuity pathway analyses on these 3 data sets, and we found 12 gene sets that were pathways that were regulated in all 3 of these data sets. The most profound one being retinoic acid signaling, which Dr. Laezza and I have been studying for a few years now. But some of the other ones were, not surprisingly, G protein coupled receptor signaling, cyclic AMP signaling, and one that we had never heard before was sphingosine 1-phosphate signaling. And so we wanted to go about finding new targets. And so we looked for the G protein coupled receptor that had the most selective enhancement in the nucleus accumbens shell. And that turned out to be GPR12. We were a little disappointed at first because there's very little information on GPR12, which makes it a good target for the Illuminating Druggable Genome project, but not great for submitting grants. But because of this R03 mechanism, we were able to study GPR12. And so GPR12, there's a little bit of evidence in the literature suggesting that GPR12 is a sphingosine 1-phosphate receptor. And so on the right-hand side, you can see this is a custom pathway where we've added GPR3, GPR6, and GPR12 into this pathway as sphingosine 1-phosphate receptors. And you can see the genes that have orange outlines, GPR6, GPR12, sphingosine 1-phosphate receptor 1, protein kinase regulatory unit, phosphodiesterases, these all show enhancement in the nucleus accumbens shell. And so the colors of the symbols, green means that the mRNA was downregulated in environmental enrichment. The red means that it's upregulated. So GPR12 is upregulated by environmental enrichment. It's also regulated by cocaine. I couldn't put that on the same figure here, but GPR12 is regulated by cocaine, regulated by environmental enrichment, and is really enhanced in the nucleus accumbens shell. So that's what we got really excited about it. My lab focuses to a large degree on behavior. That's my background. And I've always looked at reinforcement, drug reinforcement, but one thing we've been focusing on lately is frustration behavior. We found that just like humans, rats, when they get frustrated, humans, when you get frustrated, you're waiting on an elevator, the elevator doesn't come, you push the button, you hold it down longer, you hit it repeatedly. Or when the batteries die on your remote control, you push the button down longer and hold it longer and hit it harder. Rats do the same thing on their bars when they're bar pressing for drug. Panel A, you can see under FR1, where it takes 1 bar press to get an infusion. Rats have an average duration of about a quarter second. If you increase it to an FR3, where it takes 3 bar presses to get an infusion, you get an increase in bar press durations. When you bump it up to an FR5, you get a further increase. You can see the bar press data for all the animals for each bar press across panel B. The bottom row is all rolling averages of 10 bar presses, because not every bar press is a long bar press. And so we look at the rolling averages, and that'll be the bottom row. When you put rats under extinction, where you turn off the pump and they can no longer, they can still bar press, they still get the light cube, but they don't get drug. You can see that the durations go up in a very profound way. And you can see that the frustration level builds across the session. There's peaks and valleys, but in general, it builds across the session, and that's why the overall average duration is higher. When you put them under a progressive ratio is each subsequent infusion the animal has to work harder for. And this is a measure of how hard is an animal willing to work for the drug infusion. First infusion takes 1 bar press, second one takes 2, next one is 4, then 6, then 9, 12, 15. And it gets up to the point where they get their 30-second infusion. It takes over 1,000 bar presses to get a single infusion. For fentanyl, the rats are unwilling to work that hard. So at some points, the rats just decide, this is too much work, I'm gonna give up. And what we find, not with every rat, but with a majority of rats, we find, if you look at panel I, you'll find that there is a spike in bar press durations. These are rolling averages. And there's a spike in bar press durations that happens right before they give up. And so this tells us that this frustration measure is really sensitive in progressive ratio, and that's what we're going to focus on today. We also show that if you put animals under extinction for 2 hours and then you put them under an FR1, when you relieve the frustration, their bar press durations go right back down. So this is brand new stuff. Looking at bar press durations as a measure of frustration. We're still doing additional stuff. One of the paradigms that we just started was a frustrative non-reward paradigm where it's based on a point subtraction that's loosely based on the human point subtraction aggression paradigm, where rats have to get 20 points to get an infusion or a pellet, in this case sucrose pellets. It takes 20 points, each bar press is a point. There's 3 cue lights, there's 2 cue lights and a house light in the cage. When they press the bar 5 times, 1 cue light goes out, 5 more times, the second cue light goes out, 5 more times, the house light goes out, so they're in the dark. 5 more times, they get their reward. So they can kind of have a rough idea of how many points they have. Rats can't count the way we do with humans, but with the number of lights, we can see how, they can tell how many points they have roughly. And we find that with this, when we don't add any frustration into the system, it's pretty consistent across days. You can see the correlation 0.78 is pretty good. Panel B shows that this frustrative non-reward paradigm is sensitive to the size of the reinforcer. 1 pellet versus 4 pellets. 4 pellets produces shorter average durations. And more importantly, when you add the element of frustration, when you start taking away points and turning the lights back on, making the animals work harder, what we find is that medium frustrative non-reward shows the frustration scores show an inverse correlation with number of infusions on fentanyl progressive ratio, meaning that the animals are not willing to work as hard, meaning that frustration causes, or frustration is correlated with braking and progressive ratio schedule. And so we're really excited about these data. What do these have to do with GPR12? We made a viral vector, an AAV vector that expresses a short hairpin RNA to knock down expression of GPR12. We injected this in the nucleus accumbens shell, which is where GPR12 is expressed. And we did acquisition, we did maintenance responding, we did dose response, we did extinction, we did progressive ratio. And so I'm going to show you the progressive ratio data here first. Our original hypothesis was that GPR12 shRNA knockdown would decrease the number of infusions. You can see on day 1, we got a good trend. Statistically, it did not come out. On day 2, another trend that did not quite come out statistically. Day 3, it kind of washed out. If you do the ANOVA across all 3 days, you get a trend, but it's not statistically significant, which was a little disappointing. But when we looked at the average duration, bar press duration data, the frustration data, you can see on day 1, we get a trend for an increase in frustration-like behavior with the GPR12 knockdown. On day 2 and 3, the frustration behavior, the bar press durations are significantly higher. When you do the ANOVA, you do get a main effect of viral vector. which suggests that we're getting a trend for a decrease in the amount that the rats are willing to work for the drug, but we are getting a significant increase in frustration behavior. This is the first evidence we found of being able to manipulate frustration behavior in specific brain regions, showing that the nucleus accumbens is involved in frustration behavior, and that GPR12 is somehow involved. This is very preliminary, early data, really just showing validation. We also show that when you put the rats under extinction, we don't get a difference in the number of bar presses during extinction, but we do get an increase in average duration that is statistically significant. Again, showing that GPR12 is somehow, the neurons that GPR12 is expressed in are somehow involved in frustration behavior. And so because we did not get a statistically significant decrease in bar press and number of infusions, the question is this really an effect? I'm going to turn the stage over to Dr. Laezza, who's going to talk about the electrophysiology data that do support and do match with the behavior data. So, Dr. Laezza.
Yeah, thank you so much, Dr. Green and the audience for allowing me to present the data collected as part of this R03 award from my lab. Dr. Green and my Dr. Green laboratory and my lab are really contiguous. So we do a lot of things together, and our expertise is really complementary with each other. So what we are showing here in panel A is our voltage clamp, current clamp electrophysiology recording in whole cell configuration from medium spiny neuron in the nucleus accumbens shell. So those neurons either express in black a viral vector with a scramble RNA, shRNA, or a viral vector expressing the shRNA targeting GPR12 in red. And so you can see from the representative traces in the respective representative traces that the numbers of spikes, which represent evoked action potentials in response to these protocols shown below, are significantly different even at the visual inspection. And so what we observe and what we measured actually from panel B to panel E, just in the row below, are different parameters related to intrinsic excitability derived from panel A traces. So first of all, you can see that there is a significant decrease in the total number of action potentials that the neuron that are expressing shRNA against GPR12 are able to elicit in response to all protocols from the stimulation at the threshold level up to maximal current injection, as you can see in panel B. Then panel C is simply taking the maximum number of action potential possible and then confirm what is shown in panel B. Panel D and E refer to something called instantaneous firing frequency, which is basically, the frequency of action potential within an elicited train. So that is also significantly reduced in the as a consequence of GPR12 knockdown. There were other parameters that were affected, such as the current threshold in panel F. What it means is that it takes higher amount of current for neurons in which GPR12 is silenced to elicit an action potential, which is shown here. And also the voltage threshold was decreased, was more depolarized, with again is pointing toward a reduced ability of neurons expressing GPR12 knockdown to elicit an action potential. So there is overall a strong decrease in intrinsic neuronal excitability. There were other parameters that we measure in panel H and I, after hyperpolarization and the resting membrane potential and capacitance, and those parameters were not changed. We're really early in understanding what the molecular target, what type of ion channel might be underlying these phenotypes. But all combined, these data suggest that potentially voltage-gated sodium channels could be involved, especially because there was no change in the resting membrane potential. And I believe it's not shown in this data set. There was no change in the input resistance as well, which again is pointing toward potentially no changes in potassium channels that contribute to resting membrane potential. So now coincidentally, my laboratory is actually primary interest is in voltage-gated sodium channels. So we actually do a lot of studies in understanding how various isoforms of sodium channel contribute to intrinsic excitability in the brain, as well as in the peripheral nervous system. So if that turns to be the case, that GPR12 is linked, functionally linked, or signaling linked to voltage-gated sodium channel, it will be quite an interesting coincidence. But at the moment, we are very excited about these data. It's very strong effect size, and we are looking forward to do further testing with whole cell patch clamp, as well as with single unit recordings, combined with the behavior that Dr. Green presented to illuminate the role of GPR12 in addiction and in reward circuitry in general. So thank you, Dr. Green. I leave the stage to you. Thanks.
All right, so that just, the electrophysiology data confirms what we find with other manipulations that we've done is that decrease in excitability of the cells matches with the protective phenotype in the behavioral model. So it matches the electrophysiology shows even stronger that GPR12 is involved. So thank you.
Thank you. I would encourage the attendees to please place your questions onto the chat box or the Q&A. And in the meantime, we're going to move on to our next speaker, who is Dr. Daniel Isom. He started his career at Case Western in biochemistry and chemistry. He then earned his PhD in biophysics at Johns Hopkins University, where he studied protein electrostatics with Bertrand Garcia Moreno. He moved on to University of North Carolina for his postdoctoral training in Henrik Dolman's lab, where he investigated how protein dynamics are regulated by cellular stress and changes in intracellular PhD. And he's currently an assistant professor at the Department of Molecular and Cellular Pharmacology at the University of Miami, where his lab specializes in preclinical GPCRs and RTK drug discovery using informatics, robotics, and novel cell-based screening technologies. And they have a lab motto of actionable intelligence through informatics and scalable functional assays. Love it. And today we'll be hearing how he applies his research to the understudied targets by his talk illuminating how dark GPCRs detect protons. So I think you should be able to share.
There we go.
Perfect.
Can you hear me? Can you see me?
Yep.
All right. Let's see. All right, yes, actionable intelligence. That word I use all the time. You'll see it in this talk. We love it. Thank you for the invite, Anna. I see some familiar faces in the audience. It's an honor to be able to show some of our recent research to you. So the title of my talk today is Illuminating How Dark GPCRs Detect Protons, and it was definitely inspired and catapulted by our R03 support a few years ago now. But before I start, I'd like to thank my team. We are small but mighty, a mighty team of graduate students, Nick, Jeff, and Jacob, and our computational chemist, Dr. Santiago Villar, and of course, our support with which none of this would be possible. So what do we do? My research program areas are essentially the molecular and cellular basis of proton sensing with an emphasis on GPCRs, but we're agnostic. Yes, we'll do ion channels and kinases. New technologies for ligand and drug discovery, again, focused on GPCRs, but again, agnostic. And we use synthetic biology and data science to essentially tie all of this together to try to make new discoveries and push fields forward. So what I'm going to be talking about today is integrating data science with experiments. We like to do this, do some computations, test them out experimentally, see if we were correct. If we're correct, hooray, publish. That's what I'm going to talk about today. I'm going to take you out of your comfort zone with a brief overview of proton sensing. I'm going to then tell you about proton sensing by dark, pharmacologically dark GPCRs, GPR4, 65, and 68. And if I have time, I'm just going to touch on proton-gated coincidence detection by GPCRs, a whole new concept coming out of the group. So where do we start? How about with some biology? Coincident proton signals pervade biology. When we have tissue acidosis from inflammation, when we have acidification in the tumor microenvironment, at synapses, and of course in processes such as endocytosis. These are just select examples. Coincident proton signals also pervade cells, different organelles. I've already mentioned the endocytic pathway, but we also have the trans-Golgi, and we really don't have very accurate measures of how these pH values change kinetically with time. So this is still really an open box. But the important thing I need you to take home today is that all of these pH changes, which are clearly evolved given the cell has organelles with these specific instructions, are mediated by ionizable residues. The proteins have no other choice, cells have no other choice. The only way for a protein to interact with a proton signal is through these side chains. All right, and we're all very familiar with histidine being a proton sensor because its pKa value is very near normal physiological pH, if you will, at 7.2. This range here is supposed to be our operational physiological pH range from about 7 to 8. And our acidic residues, asp and glue, have pKa values that are at one extreme. And our basic side chains, lyse and arg, have pKa values are at the other. And the end product of this is that normal physiological pH, the basics are almost always positively charged, and the acidics are almost always negatively charged, and histidine can be positive or neutral. And this is why histidine is so popularly considered a pH sensor. But you can actually turn lyse and arg and asp and glue into really, really good pH sensors if you shift their pKa values into the physiological range. So we're going to upshift an acidic group and downshift a basic group. And there's a whole host of proteins that use this mechanism. It's just not very well known. And these pKa values and these residues are much better proton sensors than histidine, and I'll touch on that in a minute. All right, so here's an example, an experimental example from way back in my early graduate career. I took the side chain glutamic acid and I experimentally mutated individually into 25 internal positions in a model protein we used for protein folding studies called staph nuclease. This was done at Hopkins under the direction of the Garcia Moreno lab, as Anna mentioned. And so here's what happens. When you put these glutamic acid residues in all 25 of these internal positions and experimentally measure their pKa values, you see that a couple have the normal glue pKa value of 4.5, but most of them have an upshifted pKa value. This means that the protein is forcing the acidic group to be in the protonated neutral state. So you can get some massive pKa shifts just by burial of these acidic groups from 4.5 to 9. This is pretty huge. And notice that most of these pKa shifts are within the normal physiological pH range. So we've taken this acidic group and shifted its pKa value into the physiologic wheelhouse in some model protein where this shouldn't be possible. So it just goes to show you the power of how burial shifts pKa values into the physiologic range. You can review this topic here, but the last point I want to make is this is really difficult to predict computationally. It's really difficult to look at a buried ionizable group, lice, ARG, glue, asp, and say, this is its pK value based on some structure-based computation. This remains to be a major challenge, so you need to do experiments. All right? So when you take a step back and look at the really big picture then, and you look at over, this was done 5 years ago, 15,000 non-redundant protein chains, you find that buried ionizable groups, asp, ARG, glue, and lice, are super rare. So this just gives us the hypothesis that when you find these things in proteins, they're probably important evolutionarily. You're not going to hold on to something you don't need, and they're energetically unfavorable. So they must be important for something to end up in the core of these proteins. And one of the reasons that they are so important is with these large pKa value shifts, they have a ton of Gibbs free energy. So all you do is take the delta pKa value you have, in this case it was 3 units times 1.36, and these groups pack a punch. This is why histidine is not really the greatest pH sensor, because its pKa value is 6.5, the normal physiological pH is 7.2, 6.5 minus 7.2 times 1.36. Not so much free energy to move parts of proteins around and really create sensors that respond to pH. So we've been in the business of trying to find these buried ionizable groups and see if they're important for protein function using a program that I developed called pHinder. And this is data science for ionic features in proteins. We look at ionic features other than buried ionizable groups, but today that's going to be my focus. In a nutshell, what these algorithms do are we triangulate all of the charges in a protein. In this case, this happens to be a G alpha subunit. Then we use surfaces to discriminate what portions of these networks are exposed and what portions of the networks are buried. And once we've identified the portions of the networks that are buried, we know we've identified buried ionizable residues. And this is our actionable intelligence. When we find these, we're pretty sure they're important for protein stability, function, signaling. And we've shown that looking at 112 structures of G alpha subunits. Buried ionizable residues are important for their pH sensing. Look at 885 small GTPases, same deal, very important for their stability. Looking at 124 microbial proton pumps, we find that we have this nice conserved channel where these things pump protons and salts. But what we're here to talk about today are GPCRs. So very recently, we did a structure-based comparison of all available GPCR structures. This was earlier in 2020 when there were 416 human GPCR structures available. And so we align all the structures, we do our pHinder calculations, we find the networks, and then we cluster the results only for buried ionizable residues throughout all of these structures. And what you're seeing in these colored clusters are just where different clusters of ionizable groups were found. Their identity for the purposes of this talk isn't important. I can get into those details offline. But the important part is that yes, there are spatially conserved patterns of where we find ionizable residues in GPCRs, which is important. It's strange. You shouldn't be finding them here. This is in the inside of a membrane protein. There's water in there as well, I'm sure. What I want you to really focus on, though, is that we see ionizable residues conserved in what's going to be known as the dyad region in this talk. But I want you to notice that this spot right here, this apex site, is empty. So it's here. There are no ionizable residues that show up when we look at 416 of these structures. Another thing I wanted to point out is that we oftentimes see sodium, which is a known negative allosteric modulator of GPCRs, and it's oftentimes associated, well, it's always associated with this dyad of buried asp residues from TM7 and TM2. Okay, so what we did was we then looked at the known proton sensors. So this was what we were going for in the first place, essentially see what all of the GPCR structures told us. Now let's look at homology models. We had to work with homology models for the known proton sensors because there aren't crystal structures. There are 3 of these proton sensors, GPR 4, 65, and 68. Here's 65, here's 68, here's 4. And what we found was that in this site here, a residue pops up. We call it the apex residue, and it originates from transmembrane helix 4, whereas this dyad originates from transmembrane helix 2 and 7. And this is an asp, and a glue. I have the receptor kind of turned sideways. It's kind of a funky view, top and bottom, so that you can see that this triad forms this triangle. So the take home from this slide is that all 3 of the proton sensors have this triad. I've also indicated some additional sites, mostly histidines, that have been studied previously in the literature. You know, for the past 20 years, it's been thought that these histidines are responsible for the proton sensing function of these receptors. And I'm going to update that view in a bit. So having made this observation, computationally looking at large numbers of GPCR structures, we came to a hypothesis. And the hypothesis was that when you bury acidic residues, typically you upshift their pKa values. So we hypothesized that one or more of these triad residues had their pKa values upshifted from the normal value of 4.5 to about 7. And the way this works out in the model is as such. So the predicted model, at high pH, like above 7.5, we expect that all 3 residues of the triad will be negatively charged and associated with a negative allosteric modulator, sodium. And that intermediate pHs, we're going to kind of have a mix of protonation states, maybe a little sodium, and have some anionic states. But then at very low pH, when you're fully active, because these receptors are turned on by low pH, the triad is essentially neutralized by binding protons. And so these are our testable predictions that the triad senses protons, their upshifted pKa values. And if this is true, sodium should regulate this because sodium is known to bind in this area. Just a hypothesis. Phenomenologically, how we see this change actually happen in an experiment is that when we neutralize these groups, we will shift the pH 50 of the pH profile to higher pH values. So that's how we'll know we're correct. All right, so how do we do this? What we want to do is we want to swap out the ionizable residues that are in the triad, the asp and glue, with their neutral mimetics, D and E. So asp to acin, glue to glin. And the power of this, the elegance of this, is that we're only changing one atom. Electrostatics are on when we have the asp and glue, they're off when we have the acin and glin. So we're preserving sterics, we're not going to alanine, we're not causing any packing defects. So what we're really doing here is interrogating the electrostatics as cleanly as possible in modern biochemistry. And so Jacob Rowe spearheaded this effort, a fourth-year graduate student in the lab, and he used 2 model systems. He used our yeast model system, which we've reported on in the past, and the mini-G Brett system, which we've also reported on in the past. The way that the yeast system works is that we use high-throughput CRISPR to integrate a human receptor into the yeast genome. This receptor gets expressed. We also express a human yeast G-alpha chimera. These 2 couple, and when the receptor is activated, we produce M-turquoise-2, so the cells turn cyan. We have 10 different versions of these strains, one for each type of G-alpha coupling. So when Jacob does his experiments, he tests each proton sensor against each possible G-alpha coupling. There are no beta-arrestin experiments in this model. For his mammalian experiments, what he used were RLUC8 luciferase tagged receptors. These, when activated, these receptors recruit a venous tagged mini-G protein, and you get a bioluminescence resonance energy transfer and Brett signal. So these are the 2 models we're going to be talking about today. I think I'm doing okay on time. So Jacob was really set on doing a really big experiment where he could be sure that what he was finding was not an artifact of the experimental system being used. So there's a lot of data here. Let me break it down for you. What he did was he collected all known sites that had been mutated in the literature over the past 20 years. Now, I should say that during this process and during the process of getting the paper reviewed, XP published in the Roth lab, published a few more mutations that we do not cover here, and that some of these mutations came from XP's work on earlier studies of GPR 65 and 68, which were very helpful to us. So at the time exhaustive, but there's some new data out there. So what was the point? Well, Jacob wanted to test all of these in the same model system, same cell type, because each of the papers had done them differently, different sources, different methods. And he just wanted to make sure that there wasn't something different about these sites when compared in the same model system to these new sites in the triad that we were testing. So using this high throughput CRISPR, he developed this deep variant profiling, where in each of the 10 strains in which we have GPR4, say, he would install the D63N mutant by CRISPR, so we'd get 10 new strains. And in doing this, he created 390 mutant strains for GPR4, GPR65, and GPCR68. In pink, we have the results for the triad up top. Here's wild type, and here are all these other histidine sites which just serve as a control. And all I need you to take away from all of this data is that we're looking at increases in function of the receptor, so this is signaling, with respect to wild type of pH5, and no change or a decrease. And essentially this recapitulates all that was known in the literature, and for the first time characterizes these this buried acidic triad, which had actually never been studied before. So these data show us that in yeast, we get what we expect. Most of the receptors are functional and viable. He then switched to pH 7. If you notice, all of this work was done at pH 5 just to test the functionality of each receptor. He then switched to pH 7. And what was the point of switching to pH 7? Well, these receptors, GPR4, 65, and 68, shouldn't be very active at pH 7. So if one of the mutations that he introduced shifted the pH sensitivity to higher pH values as we predict, we would expect to see more activity at pH 7. And so what he did was he did this experiment again for all 390 of the strains and filtered out those that only had an increase or no change. And this is the subset you get out of the 390. And remarkably, it includes all 3 of the triad residues for all 3 of the proton sensors. Notice we still do pull a few of these previously studied histidine mutants through. Looking at the data in a different way, this is the total fold change of signaling relative to wild type at pH 7, and this just says the same thing in a different way, that these triad residues are really important and they're categorically different than what appears to be many of the histidine residues. Also note that in the case of GPR 65, that we don't have a pink bar here because the N, which is the conserved ASP in many class A GPCRs, really was deleterious. So in a nutshell, what this tells us is that Jacob went through this tour de force in yeast, looking at all known mutations in the literature, his new triad mutations at pH 5 and at pH 7, and arrived at a place where it looks like we're right. It looks like the computational predictions that the triad is important for pH sensing are true. But we couldn't really come to that conclusion without additional experiments. And that's when he turned to the mammalian system and the mini-G profiling assay. And I'll say that I'm just showing a representative set of select data here to make a few points, but he did well over 50 titrations looking at many, many variants. And I just refer you to the paper for all of that detailed work. Again, a real tour de force. So for each receptor, I've picked 1 point mutation in the triad. For GPR4, it's this E145Q in the apex. For GPR65, it's D286N in the dyad. And for GPR68, it's E149Q in the apex. And what I want you to appreciate is that never before, here we have the wild type curves in black, okay? Let's take a look at the most extreme example. Never before had it been rationally predicted to where you could make a mutation that would push the pH 50 or the midpoint of the pH profile so far to higher pH values, which is exactly what we predicted based on our models. And this happens in all 3 cases. So it's a very uniform response. The shift isn't as big in GPR4. We could talk about why that may be later or offline. But these results are very consistent with the pKa values in those triad residues being upshifted from 4.5 to 7. When you combine all 3 of these pink curves into one down here, what you can appreciate is that when you mutate just one of the triad residues to be permanently protonated with the N or the Q mimetic, all 3 of the receptors are function independent of pH up to about 7.4. So these receptors are already fully on at pH 7.4. And if you did an experiment over this pH range, you would never know that they were pH sensors. And this is consistent with our hypothesis that at high pH, you're fully charged with the triad negative. Of course, in the case with a mutant, one of these groups is going to be forced to be neutral. And when you're down at low pH, you're going to have the 2 groups that are neutral and then your forced neutral group. So very consistent with what we've seen, with what we predicted. To take it a step further, our predictions would say that if you removed sodium, you should see an effect where you shift the pH 50 of each of the receptors to higher pH values. And this is because potassium, if you use it to replace sodium, no longer binds in this dyad and no longer suppresses the pKa values of these groups from 7 or 8 down to the 6.5 region. And so when you lose the favorable coulombic interactions between the sodium and these dyad residues, in the presence of potassium, you just push the pH 50 to higher values. Add sodium back, you get the normal wild type curves in gray. To take it even one step further, if we neutralize the dyad with our protonated mimetic mutations, doing the D63N, D82N, D60N, D286N, we see there's no longer a response or difference between potassium and sodium, proving that sodium is actually attenuating the pKa values of the residues in this dyad. Note that yes, these mutations can be quite deleterious. I don't think this is a surprise given to where they're located in the receptor, but at least in the case of GPR 65 and 68, you can appreciate that the pH 50 is the same in both the mutant and the wild type, and that by neutralizing these residues in the dyad, you no longer get a sodium effect. We then went one step further based on our observations and thinking about the data and did a phylogenetic analysis to understand where these triad residues emerged. And if we take just a very small step back, it's been mentioned or it's been suggested in the literature that GPR 132 is also a pH sensor. We don't list it as a canonical pH sensor for a number of reasons, but one is this: at least evolutionarily, late in evolution, this is a phylogenetic tree of all GPCRs here. GPR 132, also known as G2A, came first. Then came GPR 65, followed by GPR 68 and GPR 4. When we used the mini-G protein assay to test GPR 132, we really didn't see any convincing pH dependence. So we really don't think we have pH sensing in this branch of the phylogenetic tree, and that it emerges here. And to further support that, when you look at homology models of GPR 132, 65, 68, and 4, you see that GPR 132 only has this dyad. It's actually an EAD because there's a glutamic acid residue there. It doesn't have the apex site. In fact, the triad emerges in GPR 65 and is inherited by GPR 68 and GPR 4. Also notice that GPR 68 and GPR 4 have many more histidines than GPR 65. this proves that GPR65 didn't need all of these extra histidines to be a robust pH sensor. The last point I want to make is that Jacob also went in and added the apex residue to try to turn GPR132 into a proton sensor, and that did not work. So he really tried to cover all bases here to come to the cleanest possible conclusion for, albeit a complex topic. So bringing this all together, if we want to update our understanding of proton sensing by GPR4, 65, and 68, it was initially thought, well, at least for the past 20 years, that the extracellular histidines were responsible for proton sensing by these receptors. But when you count the number of extracellular histidines, this is another data science informatics experiment, and here are the number of cysteines on the left-hand side, you find that yes, GPR4 and 68 have a lot of histidines, but so do some other receptors that aren't pH sensors. And also 65 does not have as many histidines as GPR4 and 68. It has the same number of histidines as many other receptors. So histidines are not a good predictor of what receptors are going to be proton sensitive. Instead, what is a great predictor, at least for this family, is the fact that they share this catalytic triad. This catalytic triad appears to be fully neutral at low pH when the receptor is fully active, a mix of neutral and charged with sodium at intermediate states, and fully anionic and sodium-bound NAMing at very high pH values. So this is this added level of understanding that this study and Jacob has added to this field that's really been active for the past 20 years. So he's done an incredible job, I think, in really illuminating mechanistically like how these things work. So in summary, proteins can sense physiologic pH through a variety of ionizable side chains, not just histidine. A baried acidic triad is largely responsible for dark GPR4, 65, 68 proton sensing within the physiologic pH range. Proton sensing began with GPR65 and was inherited by GPR4 and 68. And something I haven't touched upon, but several of our 4, 65, and 68 variants may be useful for developing pharmacological tools and maybe therapeutic inhibitors because they're already active at high pH. You don't need to change media pH to acidic values, which is not very good for the cells, if you wanted to do some inhibitor screening. And lastly, all 3 dark GPCRs are coincidence detectors of protons and sodium. And so I just want to take one more minute to add to this concept of coincidence detection. You might ask, well, then how prevalent is proton sensing within the human GPCRome? And based on our work thus far, we think that the type of proton sensing we see for 64, 60, 65, 68, and 4, where you're activated by a proton, is relatively rare. We do see some other instances, Adora 2A, which we're just about to publish. But for the vast majority of receptors, what we see is that once they're activated by an agonist, that protons actually gate this activity. And here's an example of that for the dopamine receptor DRD4, where we have dopamine agonism here. It's very active at pH 7, but as you drop pH, you see a cooperative Boolean-like drop in signal output. My last slide is that we see this Boolean-engraded proton-gated coincidence detection for a variety of receptors. Tom talked about S1PR1. We should probably talk about GPR12 offline after this, but I wanted to point out just to close that we also see graded behaviors, and this is another one of our dark GPCRs that we've studied in the past, HCAR3, which we're still learning a lot about. And with that, I'll stop. Thank you very much.
Wonderful, thank you. Excuse me. I encourage anyone from the audience, please enter questions, comments that you want to have via the chat or via the Q&A box to the speakers. So you were just hinting at, so is there a potential of the GPR12 having a similar triad, or have you looked at that?
So GPR12 probably wouldn't have a triad. We can look. What we do is we line up homology models sometimes against the known structures. So that's what we would have to do. But the triad is rare. It really was only seen in 465 and 68. So in this case, it would just be whatever mechanisms S1PR1 and S1PR2 may be using to gate the activity of these ligands. But I mean, we have a lot more to say on this topic. I mean, we're just getting started. This is kind of the tip of the iceberg.
I do see a question just coming in from Kanan. Is the catalytic triad found on other proteins and functional sites?
I don't know. It's as far as human GPCRs, in fact. So very specifically, as far as human GPCRs, we're only seeing it in GPR 4, 65, and 68. But in terms of that geometry and those sites, like say in ion channels and other things, I'm not sure. We've done similar types of studies in ion channels. I don't have a group that I'm collaborating with to kind of follow up on some of those experimentally, but we do see similar motifs. Right. And I want to be very important to point out, the triad isn't necessarily interacting physically. Although the dyad residues are close, it's just a feature, a pattern that's predictive of this behavior.
Right. An additional question. Do you have evidence for these buried ionizable residues to become exposed during protein conformational changes? And have you tested the position of these residues using molecular dynamics? What is the percentage of naturally occurring SMP at sites that code for these buried ionizable residues?
Yeah, so this is the question that opens Pandora's box. So using model systems, starting in my graduate years, the Garcia Moreno group has intensively studied how buried groups, when they charge, change protein structure and function by NMR, by molecular dynamics, trying to get very good at predicting these types of behaviors. And so that's a very intensive procedure and something that has never been done for GPCRs, as far as we know, which we think is kind of odd. But to answer your question, certainly when these groups are charging, I would be shocked if there's a lot of water there as well, right? These aren't truly buried. There's water inside the receptors. They're communicating with the extracellular space. So there's dynamical information is actually what is setting the PK values at what they're at. It's just we can't see it. Now the problem is it's really hard to get accurate molecular dynamic simulations of buried ionizable groups. That relates back to the slide I had that mentioned we did blind prediction studies based on all of the experimentally measured pKa values. And MD and other methods, it's very difficult for them to recapitulate what we measure. So it's a huge challenge. And if you have insights, you should look into it and maybe enter the field.
Wonderful. There's another coming from Brett. I'm sorry, amino acids are not coming up to my head, but aspartame and glutamate, is that what it is?
Asparagine and
asparagine.
Glutamine.
Thank you. Ever found to impact the interfaces on protein complexes to make complex formation pH responsive? Or do they need to be buried first before their apparent pKa increases?
Nope, they're found at interfaces. And so this is the most elegant type of experiment you can do, switch them out for the neutral mimetics.
Wonderful. For the group that's studying GPR12, do you guys have any senses of what might be a ligand for that particular GPCR?
There's evidence from the literature that it's sphingosine 1-phosphate, but there's also some evidence that possibly suggested possibly endocannabinoids.
Wonderful. I see no more questions. I think that we're going to close the session by saying thank you to all of our speakers today. Very enlightening talks. And also thank you to all the participants and attendees who have shown interest in our work. And I encourage you all to keep your eyes out for the next series coming up in fall. And thank you again for everything. Bye.
Thank you. Bye-bye.